Hi, my name is David Weinberger, and I'm going to be a senior at University School this year. I was in the medical laboratory internship program this summer, and the title of my project is Comparison of Therapeutic Drug Retrieval in BD Vacutainer Gel Tubes and Serum Tubes. There are several types of blood collection tubes available. The first kind is red, which has no additive. Then there are also gel tubes, which include the gold kind, which has a gel mixed with a clot activator, and the green, which has gel mixed with lithium heparin, which is an anticoagulant, allowing the separation of plasma. Each of these types of tubes has its own advantages and disadvantages. For example, the red tube can have hemolysis, which is when the serum mixes with the cells, and the goal of the gel tubes is to stop hemolysis from happening. Previous studies have shown that the gel in green and gold serum separator tubes can absorb drugs from blood, serum, or plasma. Several factors may affect this absorption. These factors, or variables, include the storage time, temperature, the actual drug in question, and the volume. Red tubes are the standard for therapeutic drug monitoring, also called TDM, because the effects of the gel barrier on drug levels are not fully understood. The purpose of this project was to discover, does the gel barrier affect test results? Can gold and green tubes be substituted for red ones in TDM? To test this question, I collected data on all three types of tubes. I accessed the results for red tubes and then tested the corresponding green and gold tubes immediately and at intervals of 4 and 24 hours. All of the samples were obtained from the laboratory at Hillcrest Hospital. Between testing, the samples were stored in a refrigerator between 2 and 8 degrees Celsius, and all samples were analyzed using the same Roche Cobas 6000 machine. This graph shows you the colors of analyzed tubes. As you can see, most of the tubes, the majority, was red, and this is because a red tube had to be paired with either a green or a gold, or both, to be tested. This graph shows you the different drugs analyzed. As you can see, digoxin had the most with 16 sets, vancomycin had 15, phenytoin had 8, valproic acid had 7, carbamazepine had 3, theophylline had 2, and salicylate had 1. Normally, we aim for 20 trials of drugs, but there wasn't enough time in this study to collect that much data. So for the first 4 drugs, there was enough data to produce results, and for the bottom 3, there was not enough data. After data was collected for each tube and drug type, it was evaluated using EP Evaluator software. Graphs were prepared for each combination of drug, gel tube color, which was gold or green, and assay time, 0, 4, or 24 hours. These graphs plotted the gel tube results on the y-axis and the red tube values on the x-axis. By looking at the printouts of the graphs, I recorded the slope and intercept of each graph. The 95% confidence interval standards are the accepted standards for these slope and intercept values. However, the standards at Hillcrest are a little bit less strict, and they specify that when the slope of the graph is between 0.8 and 1.2, and the intercept is between -1 and 1, the two methods of data collection are statistically equivalent. This is a sample of the graphs prepared by the program. The y-coordinates of the points are the gold tube 4-hour results, and the x-coordinates are the values for the corresponding red tubes. As you can see, if all the points were on the y equals x line, the two methods would be statistically identical. But since they're not quite identical, there's a regression line that's drawn, and by calculating the slope and intercept of that regression line, that's how we calculate results. So this is the results for digoxin, the first drug. Except for one combination, digoxin levels in gel tubes are comparable to the levels in red tubes, and additionally, the levels are mostly stable over time. For vancomycin, the data shows that the gel tube levels are mostly stable. Variations in the intercepts in the gold 0-hour and the green 24-hour may be caused from a small sample size. This is the data for phenytoin and valproic acid. As you can see, there's a lot more red than there is white, showing that the data is not quite as good for these drugs, meaning that these drugs cannot be monitored using gel tubes. Results. Digoxin and vancomycin are the only drugs that can be monitored using gel tubes. As a precaution, green gel tubes should not be used after several hours. This is because the levels of digoxin and vancomycin were not accurate at 24 hours. The data for phenytoin and valproic acid shows that drug levels decline over time, supporting the theory that the gel absorbs some types of drugs. The results of this study may not be entirely accurate due to the small sample size and the inherent logistical difficulties of such a study. For example, it's hard to keep track of the tubes and their locations throughout the lab, and it's hard to test the tubes at exact times. If this experiment were repeated again, the most important improvement would be to increase the sample size to produce more accurate results. I'd just like to thank my mentor, Joyce Lawrence, my peer mentor, Sarah Sharippa, and the Office of Civic Education Initiatives for making this internship possible.